Hi to everyone. My name is Jean-François Guégan, and I'm a senior research professor at both the French National Institute for Agriculture, Food and the Environment and the French Institute for Research on Sustainable Development. And I am also a professor at the French School of Public Health, where I teach eco-health and planetary health approaches. I am at heart both a parasitologist and a numerical ecologist working on infectious disease transmission. I worked in the past on measles and whooping cough population dynamics, cholera temporal series analysis, West Nile virus transmission, meningitis transmission as well that I still pursue to work today. And during the last 12 years, I concentrated my activities on Buruli ulcer and its disease agent Mycobacterium ulcerans. I really appreciate developing innovative research at the interface and at the forefront of many different disciplines. Within the NSF novel weapon evolution in mycobacterial pathogenesis, dispersal and ecological persistence program, I'm acting as a co-PI in charge of tasks on disease transmission using epidemiological modeling and synthesis. This program was born several years ago through discussion we had with Eric Benbow at WHO headquarters, and who was astonished to see that we were speaking exactly the same scientific language in a medical world around us, and who was much more interested by clinical aspects of disease. After my oral presentation at WHO headquarters, he came to me proposing me to collaborate, and this is what we did, really. And this is where we are. Within this program, and in close collaboration with all the people involved in, we will first use the principles of biogeography of microbes and aquatic ecology to understand both the spatial and temporal distribution of Mycobacterium species in French Guiana watersheds. What are their distribution across upstream, medium stream, and lower stream areas? And this also across different rivers that we will analyze. Let's say for a US auditor, French Guiana rivers are 5 to 10 times bigger than the Potomac River flowing near Shepherdstown in Western Virginia, for instance. So big rivers there. Aquatic and riverine animal and plant communities are strongly influenced by where these communities are located in upstream or lower stream areas and by stream orders in the river hierarchy. This is first an important aspect to consider and to take into consideration for understanding local communities of aquatic mycobacteria. This is what I have illustrated on the left part of this slide with the river continuum concept. In the medium part of the slide, an important characteristic for rivers in Southern America is that the coastal watersheds can be important, like for the varzeas on the Amazon River, and important freshwater depression zones can appear during the rainfall period in lower part of the French Guiana rivers near what we call the coastal strip near the Atlantic Ocean. After the onset of rainfall, these flooded areas can constitute ideal receptacles for the development of specific communities of aquatic animals and plants, and then of their associate microbes like the Mycobacterium. Third kind of this system is anthropogenic disturbance through activities that can modify both landscapes and wildlife community in those perturbed aquatic areas. We move to the right side of this slide with comparisons of a river catchment area without this deforestation and with deforestation. It could also be illegal mines in these areas that could impact on the ecosystems as well. We will then test the hypothesis that anthropogenic disturbance on watersheds can release in water upstreams some microbial forms that can aggregate downstream in the shallow water regions due to the current, for instance, that are also occupied by human communities. Mycobacterium ulcerans in particular will benefit in time and space from the different ecological contexts and conditions to develop and persist and coexist with human communities around and their activities. Based on previous work we produced in Cameroon, Central Africa, and French Guiana, Southern America, we will develop, with the collaboration of a postdoctoral fellow, epidemiological modeling of Mycobacterium ulcerans transmission to human, considering the different contexts and heterogeneities of local aquatic communities in the different regions of watersheds we prospected. We already showed that the probability of transmission of Mycobacterium ulcerans to susceptible individuals may exist through the biting of aquatic bugs, but that this event is relatively rare by 2.8% of probability of transmission only, that which mirrors a random process of transmission indeed. We think that this transmission pathway is passive and not active, and which show that 97.2% of transmission pathways are or need still to be explained. According to this, any contact with some aquatic animals like freshwater fishes having spines onto the top of their rays can act as a host carrier for the disease transmission as well. This can also happen with shrimps or any aquatic materials or materials hosted in freshwater system and that harbor spines or sharp edges. So the solution to the problem are numerous and will depend on the environment, its aquatic communities, and anthropogenic perturbation that can modify species composition, unfortunately, for the people living around. As shown on this slide, upper panel, bull ulcer cases are seasonal and strongly depend on Mycobacterium ulcerans flowering like bacterial blooms in aquatic ecosystem, so depending on abiotic and biotic conditions during the year. From the rainforest trees to the river to the disease in human, there are several interlinked processes that we want to better tackle and synthesize, and this will constitute our main task during the NSF program. Then, finally, and before finishing this brief presentation, I just want to mention that the French Embassy at Washington, D.C., and its Scientific Research Office and INRAE branch there will act as the focal point for US-French exchanges, partnership, and scientific collaboration, and I thank them here very much. I will end with this nice picture taken by a photographer from Radio France International radio channel during the COVID-19 pandemic in Southeast Asia. When the idiot shows the pangolin, the wise man looks at deforestation. Even if there is no formal demonstration currently on the role of deforestation in Southeastern Asia on COVID-19 emergence, let us remember that land use changes and modifications like deforestation, for instance, really constitute the engine or the motor responsible for disease emergence. I thank you very much for your attention. Bye-bye.